All right. Welcome to the U54 Partnership Research Design and Analysis Seminar Series. I'm Dan Gunderson. I'm one of the methodologists for the RDAC. Today we have two presenters, Dr. Wendy London and Dr. Clement Ma, who together are going to present introductory materials about phase 1 trials in oncology and do a demonstration of a new tool called DEDUCE, which facilitates comparison of the operating characteristics of phase 1 trial designs. Before I introduce the speakers, a couple of housekeeping items. First is that the seminar is being recorded, so that's just for your awareness. And we do that so we make it available on the partnership website, as well as through other channels, emails, and so forth. Secondly, we're going to take all questions at the end. And there's two ways you can ask questions. One is you can use the chat function. And if you use the chat function, I'll do my best to ask your question on your behalf, and I'll mention who is asking the question, or you can use the little hand raising reaction in the Zoom box, and then I'll try to facilitate your turn to ask a question. So the speakers today is Dr. Wendy London. She's an associate professor of pediatrics at Harvard Medical School, the director of biostatistics in the Division of Pediatric Hematology Oncology at Dana-Farber Cancer Institute and Boston Children's Hospital. She's also the director of the Survey and Qualitative Methods Core at the DFHEC and is one of our co-leaders for the RDAC in the partnership. RDAC stands for Research Design and Analysis Core. Her research focus is on neuroblastoma, phase 1 and randomized phase 2 and 3 clinical trial designs, identification and application of prognostic factors, and a few other things that would take too long to mention probably. Dr. Ma is an independent scientist, biostatistician, and co-director of the Biostatistics Core Service at the Center for Addiction and Mental Health in Toronto, Canada. He is an assistant professor of biostatistics at the Dalla Lana School of Public Health at the University of Toronto. And prior to that, he was right here from 2015 to 2020 as the lead biostatistician at the Dana-Farber Boston Children's Cancer and Blood Disorders Center, as well as an assistant professor of pediatrics at the Harvard Medical School. His research focus is on developing and implementing innovative statistical methods to improve the accuracy and efficiency of clinical trials. So with that, I'll hand it over to our speakers to get us going.
Thank you very much, Dan, for that very kind introduction. Today we'll be doing a general overview of adaptive and rule-based designs for phase 1 clinical trials. Clement, next slide, please. And we'll be looking at 3 particular adaptive designs, the Bayesian optimum interval design, or the BOIN, the continual reassessment method, the CRM, and then one new design that Dr. Ma has developed called the TARGET-CRM in application to a trial. And then Dr. Ma will give us a demo of the DEDUCE app, and the participants will be able to do hands-on to try out the app. Next slide. So in a phase 1 trial for cancer, we're attempting to determine the maximum tolerated dose of a new drug. So this is different from phase 1 trials for non-life-threatening conditions. So we are attempting to maximize the dose in hopes that that will maximize the efficacy of the drug. And it may be a new drug for adults, and in pediatrics, we may be attempting to determine a pediatric dose for a drug that's already being used in adults. To identify the maximum tolerated dose, we're going to look for dose-limiting toxicities in the patients. These are toxicities that are fully specified in the protocol, well-defined in advance of activating the trial. So it's very objective in counting a dose-limiting toxicity. So from now on in the presentation, I'm just going to call it a DLT, and I'll call the maximum tolerated dose an MTD. So this graph portrays the assumptions that we make that as dose increases, we hope that the efficacy of the drug will increase. And unfortunately, it's our expectation that the toxicity will also increase. There is a level at which drugs are defined as unsafe, and the investigators for a given trial will pick a threshold. Typically, the target toxicity level will be set at 20 or 30%, and above that, a drug would be considered unsafe. And that proportion is the proportion of patients that we would estimate would have a DLT at that particular dose level, or the DLT rate. Next slide, please. So let's talk about rule-based designs. For example, you may have heard of a 3+3 design. That's probably been the most widely used design to determine MTD over the last several decades. And within pediatric oncology, the rolling 6 has become predominant in the last 10 years or so. So these types of designs are easy to understand and implement, but they're very conservative because they go up carefully in terms of escalating dose. And so that's also a disadvantage because it takes a while to have enough patients to escalate the dose. And many of the subjects may end up being treated at doses that are lower than what we would need for efficacy. A rule-based design only uses information from the current dose in order to make a decision about escalation or de-escalation. We call that memory lists, and it ignores all the other information from the prior doses. So we'll talk about adaptive designs that take into account all information across all doses. Next. So here's a schematic for the 3+3 design. At the start of the trial and in the protocol, we would specify a period of time over which to observe a patient for a DLT. So it typically would be 1 cycle of the drug or 2 cycles sometimes. And so we enroll the first 3 patients and we begin to observe them to see if they have a DLT or if they reach the end of the DLT observation period without experiencing a DLT. Then we count the number of patients that have had a DLT. If there's no patients, then the model recommends escalation to the next dose level. If there's 2 or more out of 3, then we just terminate the trial. So what it says here about the previous MTD is not relevant if we've just started the trial with 3 patients. So oftentimes investigators will have to go back and amend the protocol to add lower dose levels so that they can explore what the DLT rate would be at the lower dose levels. But let's just assume for now, for this example, that we've enrolled our 3 patients and that we don't have more than 3 DLTs. Let's just say there's 1 out of 3 patients with a DLT. We don't have enough information at this dose level. So we're going to enroll 3 more patients at the same dose level. So now we have a total of 6, including the 1 DLT that occurred in the first 3 patients. So if we don't have any more DLTs, that's a recommendation from the model to escalate to the next dose. If we have 2 or more out of 6, this is the sign that we should terminate the trial and declare the previous dose level, lower dose level, as the MTD. So you can see how this would continue in a cyclic fashion until we have gone to the highest level that we can, come back down 1 to declare that the MTD. Oftentimes, a phase 1 trial will enroll additional patients at the dose level that's been designated as the MTD. And we refer to this as an expansion cohort. And this gives us more information to more accurately estimate the DLT rate at that dose level, and also may give some very preliminary information about the efficacy of the drug. So the MTD is often the level of dose that we carry forward for study in further trials. And if so, we begin to refer to it as the recommended phase 2 dose, or the RP2D. Next. So let's look at an example. Most trials make use of just 1 drug, testing escalating doses in that 1 drug. In this example, we're looking at 2 drugs, and we're going to alternate which drug is escalated. This is a trial conducted by Reed, as published in Cancer Medicine in 2016. The patients were children with refractory recurrent solid tumors. 12 patients were enrolled. All 12 of them either had a DLT or completed the DLT observation period, so which is what makes them evaluable. If a patient drops out before the observation period is completed, they're considered inevaluable and are replaced in the study. And in this case, all 12 patients were evaluable. So Reed and colleagues were evaluating 3 dose levels. Next. So here is a summary table that was published in their paper. So over here are the 3 dose levels, and you can see we began at topotecan at 1 milligram per meter squared and sorafenib at 150. At dose level 2, we escalate topotecan to 1.4, and we keep the sorafenib constant at 150. When we escalate to dose level 3, the topotecan is held constant and the sorafenib is escalated to 200 milligrams per meter squared. So in running the trial, we enroll the first 3 patients, and Reed did not observe any DLTs in those 3, so they escalated to the second level. They enrolled 3 patients, one of whom had a DLT, so they enrolled 3 more. Those 3 did not have a DLT, so now we have 1 out of 6. They escalated to dose level 3, where they observed 2 DLTs in those 3 patients. And at that point in time, they declared the previous dose level 2, as the MTD. Next. So this 3+3 design has limitations in terms of estimating the DLT rate. So they picked dose level 2, and we've got 1 out of 6, which is a 16% DLT rate. And due to the small sample size, we can see that the 95% confidence interval goes from 0.4 to 64%, very wide confidence interval. And this is really tells us next to nothing about what the actual DLT rate is for dose level 2. Next slide. Another limitation is the inability of 3+3 to go back down and gather more information. So we went up, escalated from 1 to 2, we escalated from 2 to 3, and as far as the 3+3 design goes, you stop right there. It would have been nice to be able to de-escalate and gather more information at dose level 2, or to be able to go back up and gather more information at dose level 3. So we really aren't sure that dose level 3 is unsafe. We could have just gotten unlucky in observing 2 patients with DLTs out of the first 3. Next. Additional limitations of the 3+3. In simulation studies, it's been shown that the 3+3 is able to identify the true MTD in only 19 to 41% of trials, whereas the true MTD can be identified by the continual reassessment method, an adaptive design I'm going to show you in a minute. That design can identify the true MTD in 25 to 72% of trials. And as I mentioned before, the 3+3 is conservative and ends up treating fewer patients at what would thought to be a higher, more effective, more efficacious dose. Next. Now let's look at 2 examples of adaptive designs. So the FDA guidance says that adaptive designs are an adaptive clinical trial will evaluate a treatment by observing the outcomes and then modify the trial conduct in accordance with what was observed. It's important to note that the rules for adaptation are pre-specified in the protocol. So it's not subjective. It's very objective because the adaptation is controlled by decisions that have been made in advance. Adaptive designs are just a tool. I want to point out, this is really important to know, that the computer and the statisticians are not dictating how patients be treated. The power lies with the investigator to decide whether or not to escalate, de-escalate, or stay at the same dose. The models, the rule-based models or the adaptive design models, they provide a recommendation, but in the end, it's up to the investigators running the trial to decide what's the safe thing to do. Next. So the Bayesian optimum interval design has a lot of flexibility. The investigator can specify the number of patients in a cohort that are assigned to a particular dose at a given time. Usually it's 1, 2, or 3 patients. The investigator gets to specify the total sample size. And if there are more patients, they're more likely to identify the true MTD. The investigator has flexibility in choosing the target toxicity rate. As I mentioned before, either 20% or 30% are pretty typical choices. The Boyne design is more likely than the 3+3 to correctly select the MTD, has a lower risk of overdosing patients, and allocates more patients at the MTD. Next. So here is an example of a table for the Boyne design. So in this example, the investigators picked a sample size of 15. They selected a target toxicity rate of 30%. And at that point in time, we have the information we need to generate this table. And it's placed in the protocol and finalized before the trial is even activated. And then after that, this table applies to whatever the current dose is that's being evaluated. And so, for example, let's just say using the first row, this is the number of patients that are being treated at a particular dose. Let's say we have 6 patients. Clement, can you use your little arrow? I don't think my arrow is showing up. Thank you. So 6 patients who are being treated at the level. And then of those 6 patients, the investigator simply needs to look at the other numbers in the column to see how many DLTs have been observed in those 6 patients. And if there's 1 patient, then the recommendation from the Boyne design is to escalate to a higher dose level. If there are 3, then we de-escalate. And if there's 4, the Boyne design recommends eliminating this dose from further consideration. So this table is agnostic to how many dose levels there are. It simply applies to each dose level at the time you're evaluating and trying to make a decision of whether to escalate or de-escalate. Next. So designs like the continual reassessment method, the CRM, make use of Bayesian inference. And this is a nice way to include the educated guesses of the investigator in specifying the prior probability. So the investigator specifies what the chance is of a DLT at each dose level. And then we conduct the trial and observe some data about the patients at the dose levels. And the model combines the prior plus the observed data and calculates the posterior probability. And these are updated estimates for, at each dose level, what the DLT rate is for that dose level. Next. So in the continual reassessment method, we use a mathematical dose toxicity model. So the probability of a DLT is equal to the dose raised to the exponent of a parameter that we can modify. So the parameter beta, depending on its value, changes the curve relationship between the dose and the posterior probability. So in the plots on the right, you can see varying values of beta and how it changes the assumption that we make about the relationship. So in this example, the investigator has selected a targeted toxicity of 20%. So we take the information on the prior, the educated guesses from the investigator, combine that with the observed data about DLTs on the trial, run the model to estimate the parameter beta, and then plot the curve. And the maximum tolerated dose is the point where this dose toxicity curve crosses the target toxicity threshold. Next. Let's do an example. So in the figure on the right, you can see the prior, and this is the investigator's educated guess as to what the chance is a DLT will occur at each of the dose levels. So it ranges from a 5% chance at dose level 1 up to a 30% chance at dose level 4. And so in this particular trial, the investigators chose 20% as their targeted toxicity threshold, above which a drug would be considered unsafe. In this trial, we're going to enroll patients sequentially. We've set up a rule that we can only increase one dose level at a time. And the dose level will escalate to a dose that has an estimated posterior probability that's closest to this 20% threshold. We can de-escalate, skipping as many doses as we want for safety. And the total accrual on the trial will be permitted to be 18 patients. Next. All right, we've started the trial. And up here in the yellow highlight, you can see that we have dose level 1. One patient has been enrolled, and that patient did not have a DLT. And so we take this information about this one patient, we add it to the prior probabilities, the educated guesses, and re-estimate what the rate is, DLT rate, at each of the dose levels. So Clement, can you flash back and forth between the two slides? And you can see that from the original prior versus now the re-estimates of the values, that the estimated chance of having a DLT has gone down. And that's because the new data that we observed was that the patient did not have a DLT. So the model on this basis recommends that we escalate. Now, even though dose level 4 is the closest to the target toxicity level, we don't jump all the way up to dose level 4. Our rule says that we can only go up one level at a time just to be safe. And so we escalate to dose level 2. And the model recommends escalating to dose level 2, and the investigator concurs, and and the next patient gets enrolled on dose level 2. Next slide. This patient also does not have a DLT, and so we update the probability of DLTs. Clement, can you flash back and forth between the two slides? And again, with this new information, you can see that the chance of having a DLT at dose level 3 has gone down. Okay, now let's enroll the third patient. Next. And this patient has a DLT at dose level 3. So you flash back and forth, Clement, you can see that based on this new information, the estimates of the DLT rate have really gone up. And so the CRM model recommends de-escalating all the way back down to dose level 1. That is the one that's closest to our 20% toxicity threshold. Okay, next slide. At dose level 1, the new patient doesn't have a DLT. So we go back up to dose level 2. That's now the closest to the threshold. Next slide. No DLT for the next patient on dose level 2. However, it's still the closest to 20%. So the next patient is going to be enrolled on dose level 2. We stay there. Next slide. And again, no DLT at dose level 2. And now dose level 3 is the closest to 20%. So the investigator agrees that we can escalate to dose level 3. Next. There's a DLT at dose level 3. And so because it's still the closest to the threshold, we stay at dose level 3. Next. And then again, dose level 3, the next patient doesn't have a DLT, and so it goes down just slightly. Let's jump ahead to the end of the study. Fully enrolled all 18 patients, and you can see that after the 18th patient, the dose level 3 was the dose level closest to our target toxicity, and that was declared the MTD. And so the investigator is likely to select dose level 3 as the recommended phase 2 dose for further study in future trials. So one thing that I'd like to point out is at dose level 3, we have 2 patients out of 11 who had a DLT. So that's an 18.2, that portion of patients is 18.2%. However, the Bayesian model estimated the probability of DLT at dose level 3 at 19%. And these are different for 2 reasons. One is because of the curvilinear relationship of that dose toxicity equation. And the main reason is because the Bayesian model takes into account the investigators' initial guesses of what the toxicity would be at dose level 3. And that's not taken into account in the actual calculation of the proportion. So you shouldn't be worried if your prediction by your Bayesian model differs from what you calculate just as a proportion. Next. So patients benefit from the use of adaptive phase designs because we can treat more patients at higher, more effective doses. We have better accuracy, and more often we are able to identify the true MTD using an adaptive design. And we have improved precision because we get to enroll more patients at the MTD to have a better estimate of the toxicity rate at the MTD. Next. So now let's look at the TARGET-CRM. This is a design that was developed by Dr. Ma, and we've applied it to a trial in pediatric oncology. Next. In general, we're able to make many possible modifications to the CRM. We have flexibility in the cohort size. We can develop an early stopping rule if the confidence interval, lower limit on that confidence interval is higher than our target toxicity threshold. Another way to modify the CRM is to make use of the time that we've observed a patient without having a DLT. That's called a tight CRM, or a time to event. And so even if a patient hasn't completely finished the DLT observation period, we can include the amount of time that they were DL-free in the model. And then now I'd like to talk about Dr. Ma's new design, the TARGET-CRM. Next. So this has been used in a pediatric phase 1 trial of a drug called ALRN-6924, developed by Aileron Pharmaceuticals. The trial is led by Drs. Du Bois and Shulman, and Clement Ma and I are the statisticians. So this is a drug that inhibits tumors with specific genotypes. So those with MDM2, MDMX amplification or gain, TET2 deletion or loss, or PPM1D amplification or gain. And patients who have these aberrations are of special interest because they're the ones that we would expect would have a response. They would be more likely to respond to the drug perhaps than other patients. And so they're special, they're rare, and we're going to label them as the cohort B patients. And in this trial, the objective is to determine the recommended pediatric phase 2 dose of the experimental drug. Next. So it's really important for us to be able to enroll every rare cohort B patient who comes along. So let's just say if we were doing this trial using the 3+3 design, if we have 3 patients enrolled at dose level 2, we're watching them. We're waiting for them to observe, to have a DLT or to complete the DLT observation period. And in the meantime, we can't enroll any more patients because that's how a 3+3 works. So if this rare cohort B comes along, that patient can enroll. And this is a real problem. The patient who might have the most benefit of the drug doesn't have access to the drug. This patient might go off and get other treatment. This patient might progress or die. And we lose the ability to enroll the patient on the trial. Next slide. So compare that to Dr. Ma's TARGET-CRM design. In this design, this rare cohort B patient is permitted to enroll on the trial and does so at 1 level below the current dose. That's considered safe. And so this increases the number of cohort B patients that are treated on the trial. It gives the cohort B patients access to a drug that might help their cancer more so than perhaps patients without the aberrations. And so this new design prioritizes enrollment of these rare cohort B patients. And one nice benefit of the CRM model is it is able to include all the patients who are treated at any dose level in the analysis. Next slide, please. So it's really important to have good communication during the conduct of these adaptive designs. It's important if a patient has a DLT to temporarily halt the accrual, because you have to find out if that DLT would result in a recommendation to decrease to a lower dose level. And the next patient that comes along can't be treated at the current dose level if the recommendation would have been to decrease. So quick communication, the sites are emailed, temporarily halt the accrual. The clinical research coordinator enters the DLT into the database. The statisticians are informed, they run the model, and then email the PI and the rest of the study team. We meet to review the results of the model, and if the PI decides to accept that recommendation and escalate, then the IRB has to approve the change in dose, after which the other sites can be emailed and the trial can be reactivated. And all this happens within a maximum of 6 business days, ideally shorter, because you don't want to lose any patients that they're awaiting to enroll. But this compares to much better than to a 3+3 design, where it can sometimes take 2 to 4 weeks to determine what the next dose level would be. Next slide. So acknowledgements to Drs. Du Bois and Schulman for leading this trial, to Hassan El-Sahaya and Clement Ma for the TARGET-CRM design, and the trial is currently still underway. Jeff Sablinski is the CRC, and Paige Cow and I are the statisticians. A big thanks to the funding from Petals for Pediatrics to allow Dr. Ma to develop this design. Next. So I'd like to turn things over to Dr. Ma so he can tell you about an application. This is his vision for comparing the operating characteristics of phase 1 trial designs, so that the investigator can select the design that's best for their disease, their patients, and their particular needs. Clement?
Thanks so much, Wendy, and I'm delighted to be here today. Here we go. So I think one of the issues that we found is that although the adaptive designs, there's definitely benefits in terms of accuracy compared to rule-based designs, they have been relatively, not well used in pediatric oncology. So there was a review of 90 published phase 1 trials in children, and the authors looked at the type of study design that was used for these trials. And you can see in this plot from their publication, the majority of the trials use the 3+3 in blue, or the rolling 6 design in orange, which are both rule-based designs. And you can see that there's relatively few trials which have used the Bayesian CRM, or another type of Bayesian model would be the EWOC design. So there's definitely room to improve here. And part of my work when I was back in Boston was really trying to think about how can we get investigators to use more adaptive designs in their trials. And we identified a number of challenges, and we had proposed a variety of different approaches to address these challenges. And today I'm going to focus on just one of these particular challenges, is that we discovered that there's not really great software that allows head-to-head design comparisons that's usable and accessible for investigators. So to that end, we have developed a web-based platform to allow these comparisons and help inform trial design decisions by investigators. So we have developed, along with Dr. London, we have developed the DEDUCE app. This is a web-based R-Shiny application that helps investigators and biostatisticians in designing and conducting adaptive phase 1 trials. So here is a screenshot of the DEDUCE app. Wendy will also include the link to the app in the chat box, so you're welcome to also test it out for yourself. And today I'll also run through a live demo. I can try to explain all the different features of the app. So there are two main modules in DEDUCE. The first is the design module. This is where when we're thinking about what trial design to use, we can use this module to specify and compare the operating characteristics for different phase 1 designs using trial simulations. And then based on these simulated results, investigators can select the optimal design for the needs of their trial. And once the investigators have selected the design for their trial, then they can use the second module, the conduct module, where they can then implement the adaptive design, and the module will then determine the recommended dose level each time a new patient enrolls. Currently, we have 3 designs available on DEDUCE, and we've covered each of these previously. So we have the CRM, the TARGET-CRM, and then as a comparison, we also +3 design. And we are currently actively working on adding additional designs to this app. Two of the key features of DEDUCE include, again, the ability to simultaneously compare multiple trial designs for the same set of simulation parameters, and the ability to dynamically generate a written report summarizing the results so that this can be easily used in your study protocol or to communicate with the study team. And before I go to the live demo, I definitely want to thank the team at Boston Children's, including Anan Chen and Wendy, and then our partners at Northwestern Mutual. For the past year, over a year, they've been providing kind of a pro bono support in terms of software development, project management, design of the app, and they were instrumental in getting that first version of the app up and running. And they have also since provided additional funding for our team to continue development of this app. And thanks also to Alex's Lemonade Stand for their support. So now I'm going to switch screens and show you the live demo. So I'm going to just stop my share here. And I'm going to switch.
I did put the link into the chat. So while you're doing the demo, people could give it a try also. I'd just like to mention that we are moving the app to a different platform that has better computational speed. And so right now, with multiple people using it, I think you may find that it's slow, but that is going to change. So please don't let that influence your judgment about DEDUCE. It'll be fast, I promise.
Thanks. So this version of DEDUCE is our currently kind of in development version. So you'll see that it differs slightly from the version that Wendy has shared in the chat box, but we're hoping to roll out some of these new features in the production version very soon. But overall, the modules are very similar. So you see on the top, we have our menu with the home tab, the 2 modules, design module and the conductor trial module. This is the upload data module is something we're working on right now. We have some information in the about tab. And then if you're looking for more detailed documentation, you can pull up the help tab, and this opens up a separate PDF file with the detailed information for using the app. So I'm going to start off with the design module. So I'm going to click up here. And what users will see, you'll see the left menu bar, which is a series of inputs that specifies the trial that you're trying to design and the simulation parameters. And after you've entered all these, you can click simulate to simulate using the parameters you specify. So I'm going to kind of quickly walk through some of the inputs. So the first is to decide which designs you're interested in, and you can select one or any number of the designs. And again, we're trying to add additional designs here for users to compare. So I'm going to just select all 3 for comparison. Next, you have to specify how many dose levels are in your trial. So this is a slider bar. You can see I can change like this, and I'm going to keep this at 4 dose levels, similar to the aileron trial that Wendy has shown earlier. And then you could also specify the label for each of the dose levels. And there is some built-in error checking. So for example, if I try to only include three labels for a 4-dose level trial, it'll give you a little error message. So I'm going to just put it back in. Next, you can specify the starting dose level for your trial. And it doesn't have to be the lowest dose level. So in this case, we're starting at this dose level 1, which is actually the second lowest dose level. And this gives us allows us ability to de-escalate from the initial dose if we encounter early DLTs. And this is quite commonly done in phase 1 trials. Next, for the simulation. So in order to calculate some of the operating characteristics of the trial, what we do is we perform statistical simulations to see on average how often we get the right answer. So the more simulated trials that you run for your scenario, the more accurate the result will be, but it will take more computational time. So for the purposes of the presentation, I'm just going to set it to 300 simulation replicates here. But we would recommend that for your actual trial, you should run at least 1,000 simulated trials. Next, we can specify that target toxicity rates for your trial. And here I've set it at 20%. And then for your simulations, because we are simulated, we can then specify the underlying true toxicity rates for each dose level. And here what I've indicated is that for example, for dose level 1, I've indicated that there's a 5% chance of a DLT. And for the top dose level, there's a 30% chance. So you can see that this dose level 3 here, which has a 20% DLT rate, matches the target toxicity rate. So this dose level is the true MTD, and that's what we're trying to find. In the simulations, we're hoping to pick this one in your simulated trials. Next, we can specify how quickly we're able to accrue participants. So this is given by the average time between patient enrollments in days. We also specify the DLT observation period in days. So this is how long you observe participants for developing a DLT. And this is typically set at 1 cycle of treatment, so usually 3 or 4 weeks. So here I've set it at 28 days. Next, for the Bayesian models, we need to specify that prior toxicity probability for each dose level. These, again, are our initial guess of what the toxicity rate might be. And this can come from either preclinical data, or it could come from a prior clinical trial. So in pediatrics, often we do have data from the adult trial for the same drug. So we can use some of that information to inform the selection of the prior probabilities. And these priors can differ from the true probabilities here. But if we're lucky and our prior is very close to the truth, then our design will be more efficient. You've already started very close to what that true vector would be. Next, we specify the maximum sample size. So when the Bayesian designs will stop the trial. For the 3+3 design, there's not a preset maximum sample size. If you remember that flow diagram that Dr. London showed, depending on the progression of the trial might end between 3 or 4 doses. Like it could have 3 to even up to 24 patients. So this parameter only varies for the Bayesian designs. We can also alter the cohort size. So this is the number of patients that we require to enroll on the current dose before we're allowed to escalate to the next higher dose. And then for the TARGET-CRM design, we need to specify in our population that we're recruiting from, what proportion of patients will have these rare tumor genotypes or tumor histologies that might be more responsive to that targeted agent. And in this case, I've specified it as 10%. So here I'm going to just keep most of these the same, and I'm going to click simulate. And you see it's going to, the computer's running the R programs that we've developed in the background. and it's going to display the results in a couple of seconds. So, and we would love for to receive any feedback. If you do get a chance to try out the app, please reach out to us and let us know what you think, things you like, things you didn't like. We would love to hear back from you. So here are the results. You can see there's 4 main figures showing different metrics that we might consider in deciding which design that we're going to pick. So on the top left, this is a metric of accuracy. So how often in our 300 simulated trials do we pick the true MTD for each design? So if you remember, given our true toxicity rates and the target toxicity rate, the this dose level 2, the third highest dose, is the true MTD. So we want to maximize the chance of getting the truth. So the higher the bar, the better for this this dose. So you can see that 3+3 design, it's able to identify the true MTD about 34% of the time, whereas the TARGET-CRM design is better. It has a higher accuracy over maybe 43% of the time. So there may be some benefit for picking the TARGET-CRM in this case. Next, we can look at how often patients are allocated at each dose level. And our goal here is to try to assign more participants close to or at the MTD, but try to restrict the number of participants enrolled at doses above the MTD, because those would be overly toxic and potentially unsafe. So you can see, it seems like the 3+3 design does a pretty good job of assigning patients at the true MTD, so that's good, but it also sometimes overshoots, and we end up enrolling a higher proportion of patients at this dose level 3, which has if you remember, has a 30% chance of getting a DLT. Next, on the bottom, we have another metric for safety. This is how often patients experience a DLT at each dose level. And you can see, as expected, as we increase the dose level, the percentage of DLTs increases, but it's still well below the 20% threshold. So this, this setting seems okay. And then finally, we can look at the trial duration. This is the mean duration in days. And you can see the 3+3 design has a shorter trial duration compared to the 2 Bayesian designs. And this is really driven by the, on average, smaller sample size that a 3+3 design would need. And on average, it won't need 18 participants, whereas the two CRM and the TARGET-CRM designs, we've required, based on the parameter we set, to have a sample size of exactly 18. So there's some trade-offs here. For the Bayesian designs, we get higher accuracy, but the 3+3 design might run more quickly. So for the investigators, this is what we want to present: a head-to-head comparison across different metrics so that people can make an informed decision. And then all of these figures and graphs, we can download it in a kind of a downloadable written report. I'm going to just show this very quickly. Kind of open the Word document here. So you can see here, there's some written summary explaining what the simulation parameters were, what some of the metrics, and how to interpret the results. You can see each of the 4 figures are listed here, along with the table with the actual numeric values for some of these metrics, so that investigators can plot additional figures or compare across different simulation runs. So I'm going to close this window. Sorry, go back to my. Okay, so that's the design tab. Next, I want to show you the conduct tab. And here I'm showing a slightly newer version. This will differ slightly from the version you'll see, but we're hoping to roll this version out very shortly. So here, the investigators have already selected the design for the trial. So in this case, we have the ability to run either the CRM or the TARGET-CRM designs. For the 3+3, because it's rule-based and it's very straightforward, we have not implemented a specific module for that design. Some of these parameters are very similar to the ones that are in the design module. And then here you enter your observed patient data. So you would enter your patients one by one. So let's say we enroll patient 1. Let's say they didn't have a DLT, okay? And then we assume that, let's say we specify the cohort size as 3 patients each. So since we've only entered 1 patient so far on dose level 1, there are 2 slots remaining in the current cohort. And so we could just run this model here. And then this generates the posterior toxicity probabilities for the different dose levels. You can see that here's our 1 patient. They didn't have a DLT. And currently the estimate for the DLT rate is 5%. And the recommendation is actually to enroll the next patient at dose level 1, to stay at the same dose. And that's because we haven't completed the cohort 3 patients yet at dose level 1. So we're not allowed to escalate yet. So suppose we've added 2 additional patients here. So now we have completed our cohort of 3. Let's say none of them have had a DLT. So now we change this to 0 slots remaining. We've completed our cohort. We can rerun the model, and we can see here now we have enrolled 3 patients, no DLTs. The posterior DLT rate is very low, only 1.7%. So the model's telling us that it's safe to escalate to dose level 2. And then this would go on as you kind of proceed in your trial. And you would, there's also the ability to download a report with all of this information. I'm just going to show this very quickly. So right here, so it shows the observed data, that DLT probability table that you've seen, and some of the kind of recommendations and some of the assumptions for the trial. So this is a report that could be used for the investigator to, or for the statistician to share with the study team, or for the investigator to report back to the ethics committee for dose escalation decisions. So with that, I think that's the end of my live demo. We would be very happy to take any questions at this time. Thanks very much for your attention.
Thank you, Dr. Ma and Dr. London, for a very interesting presentation. I certainly learned a lot, and a demonstration of a very cool tool. I thought that was really neat. A reminder to the attendees, if you have a question, you can use the chat function in Zoom, or and I will read your question for you, or under the reactions button in Zoom, you can raise your hand and I'll call on you to ask your question. And so while people get sort of situated with that, I'll start off my questions, or I'll start off questions with one of my own. And I was interested to know, and you guys, I think, answered this question a little bit in your talks. And so it's maybe a little bit down in the weed question, but for the Bayesian designs, I'm wondering, what is the process for selecting the prior? I know you can get it from prior studies with adults and things like that, but sort of since these are small sample trials, if you select, say for the beta prior, if you select the alpha and beta parameters that are too informative, then the prior is going to dominate, right, over the smaller sample size. And so, you know, where do you kind of select the alpha and beta parameter for beta distribution as your prior, given that you're talking about, you know, 20 and less participants?
Yeah, maybe I can take it, and Wendy, please fill anything. So you're exactly right. So you know, these are small trials, and the prior could be quite informative, meaning if you misspecify your prior, you know, you might start off from a place that's quite far from the truth, and it'll take you potentially more patients, more observed information to kind of kind of recover back from that. So part of it is, you know, we try to make the best guess possible based on available data. So thankfully, in pediatric trials, we typically have adult data to rely on, and obviously it's not a perfect, you know, relationship between how children might respond versus how adults might respond, but that gives us a ballpark range, and we speak to our investigators to try to pick an appropriate prior. There are some statistical algorithms that have been published to try to help investigators to pick an appropriate prior, or at least pick a prior that will not be as informative. So it will, sometimes they would specify a prior that's quite spaced apart, meaning each jump in the prior distribution, there's a larger gap between the probabilities, and that
Yeah.
allows the algorithm to explore more dose levels. You can imagine if let's say dose 2 and dose 3, the prior was 5% and 6%. Well, the algorithm's going to have a really hard time determining which dose level it's going to be. So that's another way, a statistical way to do it. And then lastly is part of the design process. When, as a statistician, when we're designing the trial, when there's simulations, we try all sorts of different priors. So we try the prior that the investigators has recommended. We try priors that might be quite different from that. And that way we can see how the prior affects the accuracy. And that also might tell us, well, if we misspecify, we have a lower chance. So maybe we need 3 more people in our trial.
Yeah.
So that's part of that decision process. And that's why we're building this tool that you can include or try different priors and see how it affects your accuracy, so that you can make an informed decision.
Thank you. I do actually have more questions, but I'll save mine for later and give other attendees a chance to ask their questions. So again, use the chat function or the little raise hand function if you have any questions. So far, I don't see any. So I'll ask my next question. We have 2 minutes left. My second question is, there's, in my very limited experience in therapeutic trials, or at least drug trials, there's always some question of what are adverse events. And some we obviously know from past studies, but some we don't know. So for example, if you have, I guess the most recent famous example would be the vaccines we have for COVID. We didn't quite know what those adverse event may be. Some of them, I'm sure they had a good idea, but that was a pretty open question. So a 2-part question, I guess. How are they measured? Are they usually biomarkers or patient-reported outcomes? And my second question is, this is not really a phase 1 question, but at what point in the sort of phase 1 to post-market surveillance do you incorporate measuring sort of potentially unknown adverse events?
So maybe I can give this a shot. So for chemotherapy, the profiles of agents and families of agents, the toxicities are pretty well known. And for the most part, hematologic toxicities are ignored because patients can recover blood counts and are expected to. And so typically DLTs include non-hematologic things that are grade 3 or higher, according to the CTC, the clinical toxicity criteria from the NCI. And so it is pretty well known. It becomes more straightforward, certainly for cytotoxic agents. Where things become a little trickier is when we get into biologics, or we get into a brand new class or a brand new family of drugs. Anything that's first in human, you're right, Dan, you won't really know what could potentially be a DLT, but things that are standard, that are anything that's life-threatening is going to be a DLT. And so even though there's a list in the protocol, there's also sort of a general category of anything that's grade 4 or life-threatening, or anything that's where a patient dies is considered a DLT. So it is left kind of wide open. And then the regulatory body requires reporting of anything that's unexpected to the FDA, or anything that's grade 4 or higher. So even right from the start, you don't even have to wait until marketing. The government is accumulating this database of everything that's known about the safety of a particular drug. Does that answer the second part of your question?
It did, yeah. And in fact, I was thinking of biologics when I asked the questions, as well as JAK2 inhibitors, which is less known what the adverse event profiles are. So thank you. We're actually a couple minutes over. if anyone has questions they didn't want to or didn't have time to ask, feel free to shoot me an email. I can pass it on. And with that,
And please email us with your feedback on DEDUCE. Clement and I would be very interested to incorporate your suggestions to make the app more user-friendly and understandable and useful. And so I guess you can contact us through Dan, but we would love to have you go online and try out the app. I sent the link in the chat.
and we can Wendy, do you want us to include the link when we send the recording notification around?
Yes, please.
We'll do that with a request for you all to contact them with feedback that you may have.
Thank you.
Very much, Dr. London and Dr. Ma, and we hope you have a wonderful rest of the day.
Thanks so much. Take care.